Welcome to this session on minor blood group antigens and antibodies. This is about screening, cross-matching, and the clinical significance of these antigens and antibodies. The objectives will be to understand the concept of minor blood group antigen systems and the role that antigen disparities or polymorphisms within them play in provoking the development of positive red cell antibody screens, and hence the potential for future acute or delayed hemolytic transfusion reactions or hemolytic disease of the fetus and newborn. When we think about what unidentified antibodies might be on a panel, we will be inclusive to define what a BG is as one of those possibilities, and generally bestow an understanding of the current technologies for antibody detection in our quotidian wet techniques with the associated benefits and limitations of the different kinds that are available. Red cell serology began at the turn of the 20th century with Karl Landsteiner's discovery of the ABO system, a carbohydrate-based system that therefore foremost provoked IgMs. IgMs being readily detected at room temperature, which is colder than core body temperature, and readily detected without the need for any other reagents because IgM itself naturally pentamerizes. Now, the majority of red cell antigen systems are protein-based, which means that they're ultimately provoking IgGs as the isotype of concern. And these, being monomeric, do not naturally produce visible agglutinates of red cells. Operating at 37 degrees, the optimal conditions, also requires incubation energy. And so the real breakthrough in being able to identify these systems by virtue of the IgGs that they provoked really only took off by the contribution of Sir Robin Coombs. He opened the door to the discovery of hundreds of blood group antigens with an anti-human globulin and anti-IgG, the Fab of which recognizes the FCs of IgGs, bringing those and the red cells that they're bound to together in a visible agglutination endpoint akin to that seen by IgM alone. And so what we saw was a clear inflection point after the broadening of the use of this reagent so as to help discover many subsequent blood group antigen systems. Though there might have seemed to be a dry spell, there was a bumper crop in 2012, and since that year, an almost annual listing of a new system, essentially a taxonomy to reorganize so-called orphan antigens and series and collections to get a cohesive understanding of how these interrelate to each other and are founded on a single base molecule. We want to acknowledge that since the fall of 2022, there are 44 blood group antigens listed to date and over 400 red cell antigens from among over 1,000 blood group coding region variants, not all of which are immunogenic, though probably more than this evident 400 are, because not every human immune system has encountered all of these coding region variants to see how immunoprovocative they might be. The ISBT catalogs in a systematic way each of these serially discovered blood group antigen systems with an ISBT number, a system name, a symbol, a listing of the known number of antigens within each of these systems, a corresponding gene name, and of course its chromosome location. There are some nomenclature issues with some of these blood group antigen systems also having a cluster of differentiation number. And there are some facts to be appreciated with respect to the epitope or carrier produced by each of these individual red cell structures. These don't just exist to create jobs for blood bankers. Many of these are functional molecules, and mutations within them, or null states, can produce some striking and clinically important syndromes of interest. The difference between a major and a minor antigen system is essentially the matter of isoimmunization as a consequence versus alloimmunization. The two major antigen systems are ABO, which associates with naturally occurring isoagglutinins. The other major system, RHD, a big 12 transmembrane pass protein. The bulk of D variation beyond the positive wild type is the negative state, which means many exofacial loops of confrontation when an antigen negative person gets exposed to D positive blood, be it in pregnancy or transfusion. So isoimmunization is a high odds event with antibodies against more than just a single SNP-based amino acid change locus. On the right-hand side are examples of alloimmunization. These are subtle changes in molecules shared between donors and recipients, but nevertheless immunogenic, significantly less so, at an odds of maybe 1 to 10% per disparate antigen exposure in a pregnancy or transfusion encounter. Now, some of these minor antigen systems typically associated with alloimmunization can associate with isoimmunization if the entire protein is missing and not just mutated. So if one is missing the entire KX protein, one manifests with congenital hemolytic acanthocytic anemia and the so-called McLeod phenotype. Rh null individuals, by missing more than just the D structure, but the RAG protein that is involved in the DCE complexes that form on red cell membranes. This necessary RAG molecule, if missing, doesn't permit any docking of Ds or CEs in the Wiener system. And so these are less stable erythrocytes as a result and can have morphologies ranging from stomatocytes to spherocytes with osmotic fragility. Glycophorin deficiency may produce an elliptocytic morphology and some cation channelopathies, such as deficiencies in the Diego system. Each of these states, if transfused with wild-type blood, are more than just alloimmunizing, but rather isoimmunizing with the provocation of high prevalence antibodies. These, therefore, are considered rare blood types. It's much harder to find similarly McLeod phenotype or Rh null red cells compared to the classic minor antigen system differences and associated alloimmunizations. Now, the Coombs reagent, or anti-human globulin testing, can encounter a blood bank specimen in one of two ways. In direct testing, if we are pipetting up erythrocytes to examine for existing immune deposits or opsonins, IgGs already bound to a host's red cell population or complement, already opsonizing those red cells. There are polyspecific Coombs reagents that can be sensitive to the deposit of either. If we're pipetting up yellow stuff and examining antibodies not yet bound to their targets, we can use informative red cell surfaces, representative decoder cells, so to speak, in the reagent red cells that are chosen in screens or panel investigations. Knowing what those antigen profiles are and seeing if antibodies bind to those red cells with proof of that with that same tool of anti-human globulins can help establish what specificities a person is attacking. And this therefore informs what antigen negative blood to source for such individuals. These plasmas from specifically sensitized individuals can be validated in a cross-match against a candidate unit, again using the anti-human globulin to prove that the antibodies of concern are indeed not landing on a successfully selected unit that is supposed to be target negative to assure a safe transfusion. The indirect antiglobulins used in screens or cross matches are concerned only with proving the attachment of IgG and is therefore monospecific, in contrast with the polyspecific Coombs reagent used in assessing for existing immune attack towards patient population red cells. There are really three key testing orbitals or philosophies within the blood bank. The nucleus, the core of our interest in testing at room temperature and immediate spin, is establishing the group or the type of the patient. So that is speaking to one's ABO type and their RHD type in terms of pos or neg. This is asking the question, with what blood types may I honorably transfuse my patient, or what can't my patient get conversely, because otherwise Landsteiner or a fetus may be violated. D seroconversion is highly important towards the possibility of HDFN. Thankfully, we have the labeling of the D attribute on our blood bags, and we can also RH immunoprophylax against this seroconversion. But if we don't heed these readily available mitigants, D seroconversion is a high odds event that can produce a high impact syndrome in RH isoimmunization disease of the fetus or newborn. The next orbital above the core of grouping or typing is screening. And that's asking the question of whether or not there are non-ABO antibodies in a patient that could threaten a forthcoming unit despite it being group suitable. It's a theoretical snapshot surmised by informative group O testing cells. We're not asking about the ABO or RHD type here, or antibodies against those structures. Our focus here in group O erythrocytes is to see what antibodies a person may have to everything outside of the ABO system. If a screen is positive, it tells us what kind of an antibody looms, and we get high resolution or specificity deductions going in so-called panel investigations. If a screen is negative, we're off to the races. We're good to go, and we need only heed the group from that core or nuclear testing with establishing your patient's group. A positive screen, again, an indirect antiglobulin test using an anti-human globulin, maps to serologic cross-matching. A screen-positive person needs to have their selected blood unit bypassing the range of their antibody specificities. And so the question of the cross-match is whether or not the proposed unit is indeed compatible. In this true-false question, is the ONAG red blood cell devoid of immunogenic antigens on its surface and therefore universally transfusible to anyone? The naive individual who is unaware of the existence of the many minor red cell antigen systems might be tempted to say that this is true, but it is clearly false. Why we screen is about the fact of our patients potentially having red cell antibodies to blood group antigens other than within the ABO RHD system. Even if you hang a unit that lacks the A or B structure of the ABO system and the D antigen that gives you your positive as opposed to your negative attribute, an alloimmunization to SNP-based variants of other red cell structures can readily attack such a so-called universal blood type red cell. There are 10 key serotypes that we can phenotype for in the blood bank. The RHCE molecule, highly resemblant of the RHD molecule. There's the Cal structure. There's the Duffy molecule. There's the KID molecule. The MNS or glycophorin molecules, M versus N expressed on glycophorin A, and the big S versus little s expressed on glycophorin B. In bold are the antigens to which we do have typing anti-sera. And so when we talk about more extended or high fidelity matches, the 10 serotypes that we're usually talking about supermatching are the C's and E's, the big K status, the Duffy antigen profile, the KID antigen profile, and whether one is big S positive or not. These are the practical antigen determinations and emulations that we can therefore prepare in giving to a patient the best possible or highest fidelity blood match, should such a justification exist. So again, polymorphisms are about sharing the same protein or thing with your donor, but acknowledging that there may be different versions thereof. One can have a big K with a little k allele. The little k host is at risk of making an anti-big K antibody if seeing a red cell expressing these antigens. If the Kell system was represented by dogs, the big K here is the Great Dane. Now, red cells express antigens, but the antigen systems expressed upon them may be differentially expressed in other tissues. And this may have implications in histocompatibility, because if the kidney also expresses the Colton, Duffy, or KID blood type system, and a host, an intended recipient, is presensitized to the intended graft's polymorphism, that may predict for a graft threat. So knowing blood group antigen systems is not just the business of blood bankers, but of potential interest to solid organ transplanters as well. The table on the left lists what solid tissues some of these antigen systems may be localized at or differentially expressed within. The bar graph is giving a bit of an outdated but still relative quantitative sense of antigen diversity and the nature of antigen expression. You could see that the MNS and RH systems have a higher number of antigens within them compared to other systems, and that all the systems in gray bars are subject to significant antibody-mediated attack. These are intrinsic to the red cell membrane. Acquired antigens in black here, on the other hand, are slippery and tend not to be the targets of clinically significant hemolytic transfusion reactions. And in fact, in the blood bank, antibodies against these structures can be plasma inhibited as a maneuver to melt away that reactivity and to therefore infer what possible systems are at play in the specificity of a given identified antibody. In white here, you could see that there are intrinsic antigens defined by glycosyl transferase genes, and that's simply speaking to the ABO system, which was covered in the first seminar. What's wrong with this phrase that came from a dictation? Quote, "It was impossible to find suitable blood units that were free of antibodies for this patient." This is a bit of an absurd statement. Yes, it's speaking to the challenge of serving a patient adequately, but the better way of saying this would be to say that it was impossible to find suitable blood units that were antigen negative with respect to the antibody repertoire of this patient. It's not the antibodies and the products that are inflicting harm upon the patient. It's the intrinsic antigen expression of erythrocytes that could make them targets of hemolysis by the patient's existing antibody repertoire. Who makes red cell antibodies, or who might have a positive screen? Well, about 1 to 10% of the population is screen positive. That's all comers, and that's not speaking to immune responder groups that have higher rates herein. But the foundations of screen positivity begin with an exposure to foreign red cell antigens, and that can be by pregnancy or transfusion, or to the scant passenger erythrocyte contaminations of solid organ transplant grafts or even tissue grafts. An exposure cooperates with the recipient's intrinsic seroconversion risk, that is, whether or not they're an immune responder. And the detection of the antibodies on a screen depends on how recent and how durable the antibody is, because about half of seroconversions will evanesce within a year. And so this produces an underestimate of the true memory B cell baggage that individuals have. If 1 to 10% of the population has a positive screen, this is essentially saying that many more have antibodies that may have evanesced. That antibody screen may be falsely negative if there was only one antibody and it's evanesced already by the time of testing. Further to screen positive epidemiology, as mentioned, 1 in 10 recipients have made red cell antibodies. It's probably more than that if we were to torture the memories out of memory B cells to ask them what exactly they've made in the past. The seroconverts, as mentioned, are also known as immune responders. And in this famous blood paper by Higgins and Sloan, what got modeled out was a responder proportion of about 13% based on what was deconstructed to be a 30% seroconversion rate for disparate antigen exposure outcomes with a 70% non-response rate. So this cycles with every subsequent exposure so that immune responders make themselves evident early on, eventually reach saturation, whereas non-responders can get transfused virtually ad infinitum and maintain their screen negativity for longer periods. This explains why there's so much diversity in seroconversion outcome literature. Some papers saying that this is a dose-dependent or exposure-dependent phenomenon, and others saying that dose or frequency of transfusion doesn't matter. Both realities hold true if you acknowledge that there are responders and non-responders informing that specific cohort. The immune responder state varies by condition, and the most rigorous of all immune responders are those with sickle cell disease. Sickle trait is also an immune responder, but other states really don't hold a candle. And so while we respect that myelodysplastic syndrome has an increased odds of making antibodies and may be of value in prophylactic matching, they have to wait in line before the priority of higher fidelity matches for those with sickle cell disease due to their extraordinary and much higher seroconversion rates. Hemolytic sequences. It's obvious that an acute hemolytic transfusion reaction is founded on the immediate interaction of existing host antibodies confronting incompatible antigens at the moment of transfusion. This is an event that is occurring within 24 hours. It can be clinically dramatic. The antibody is already present, and the antigen may be present in high density. And so the speed with which this happens is virtually inevitable. And that's in contrast with primary sensitization, which can take 1 to 3 months after a given red cell antigen exposure. The antibody is not there at first, but the immune synapse is negotiating an antigen presentation to a T cell, which eventually communicates with B cells, which eventually secrete the specific antibody provoked by the antigen that got presented. By the time the antibody titer is of sufficient concentration, the original antigen presented in the immune synapse is essentially gone. Transfused blood doesn't last for more than a few weeks, and this seroconversion, the eventual detection of the provoked antibody, occurs typically many weeks after that. And so these are clinically inapparent events, but not unimportant, because what they can give rise to are reactivation responses known as secondary sensitization or delayed hemolytic transfusion reactions. Delayed relative to the archetype of acute hemolytic transfusion presentations, but not delayed in the sense of the speed with which reactivation occurs compared to the original primary event. This is where the antibody is recalled from undetected quiescence or evanescence. This is about a booster or an anamnestic response. Anamnesia, meaning the opposite of forgetting, a fancy term for the immune system. A unit that is positive for a target antigen confronts a host whose relevant antibody may have waned, but this flashes back at higher speed and reaches a higher titer than was ever made the first time. This is really analogous to the deliberate vaccine-induced booster biology. This response can be anywhere from subtle to serious. Serologic transfusion reactions. Again, acute, delayed, or primary have their signature timelines, but you can appreciate from this picture that sometimes there are overlaps and it can be hard to distinguish a slow, delayed response from a very fast primary response. At the end of the day, what's important is how long the blood may still be there for its destruction to be evident and potentially clinically harmful to the patient. So some slow delayed reactions and some fast primaries can be equally significant. And depending on the eye of the beholder or the coder in the patient's chart, ultimately be called a delayed hemolytic transfusion reaction. Now, what are your matching options? Standard of care was basic ABO RHD matching, a two-way match. The prophylactic antigen match goes a little further. It includes the C's and E's of the Rh system, as well as the KEL antigen. KEL is quite immunogenic, a 30% seroconversion response when a little k confronts a big K positive unit. Prophylactic antigen matching is usually offered to as yet unsensitized but sensitization prone patients with sickle cell disease or those with autoimmune hemolytic anemia who have not yet made alloantibodies. The extended antigen match brings it up another level. It includes the KIDS, the Duffys, and the S match. It is important to appreciate that there are differences in antigen expression between people who have African ancestry, that is Blacks, versus those with European ancestry. One way to remember this is that the capital letters are the likelier immunogenic confrontations when a person with African ancestry is confronting a predominantly European donor base, as might be the case in someone here in Toronto with sickle cell disease. The big C, the big E, the KEL, the capital K antigen, and the big S antigen exist more often in people who identify as white as opposed to those who identify as Black. We appreciate that those who are Black tend to have a base Duffy B genetic profile, though it's often silenced by the GATA promoter mutation. And conversely, those who are not Black tend to be enriched for the Duffy A blood type. So Duffy A is what a person with sickle cell is likely to be bumping into if the blood is not appropriately matched. Opposite to that, JKB is the likelier antigen of confrontation to a person who may be Black. And so the mnemonic here, if you are working in the dark and trying to source the best possible unit for someone with African ancestry whose profile is not yet known, you probably won't do wrong to select big C negative, big E negative, KEL negative, big S negative, Duffier negative, JKB negative blood. Now, what about our testing platforms? Well, first of all, grading is semi-quantitative, and it runs from the notation of 0 for a non-reaction, a negative reaction, to a weak to 1 to 2 to 3 to 4 plus reaction strength, if positive. We have 3 wet bench platforms. The classic is the conventional tube technique using saline indirect antiglobulin testing. There is the gel microtube technique, also known as GLIAT, for gel low ionic strength antiglobulin test. The low ionic strength saline is the potentiator in the gel microtube. And then there's the solid phase red cell assay in microtiter plates, abbreviated SPERCA, or by its patented name, the capture technology. A positive reaction in the conventional tube technique is an agglutinative button at the bottom. This looks a little different in gel microtubes. The gel matrix represents an interference to the traversing of red cells with spinning or centrifugation. If the red cells are not stuck together, they can make their way to the bottom. But if red cells are agglutinating in a positive reaction, they will stay stuck at the top. The appearance of a positive on the solid phase microtiter plate is this slaking or this diffuse pink over the entire hemidome of that microplate. The negative reaction pools the erythrocytes down at the very bottom by gravity. The cross-match, usually done in tube or gel at our institution, can be spoken to in several ways. When we talk about crossing and reserving blood, for example, 2 units, let's say the corduroy and the denim unit, we can describe the nature of its cross-match compatibility by how that testing was done. Immediate spin is simply mingling the patient's plasma with samples from the candidate units at room temperature. It's a quick spin, and it's an immediate determination of whether or not there is incompatibility. And because this is at room temperature, without any additional reagents, this is only identifying IgMs, the most important of which are ABO antibodies, though the odds of catching any such interaction are incredibly low because of policies to have candidate units twice typed, that is ABO confirmed, and with current CSA standards, likewise requiring a patient to have been at least twice typed with concordance in that information before being assigned type-specific blood as opposed to Group O blood in the interim. The Coombs phase or complete serologic anti-human globulin-based cross-match really imitates in vivo conditions at 37 degrees and focuses on IgG detection with that reagent. Immediate spin and incubated full serologic cross-matches are serologic methods. You put your gloves on and you're working with test tubes or microtubes to do this work. That's in contrast with electronic cross-matching, which is inferential. It's using the information you had on your screen, and you are eligible for this only if you are screen negative. If you are screen negative, we know that all we need to do is give you ABO RHD suitable blood in accordance with matching conventions. Electronic cross-matching is assumptive and dry. You don't have to put your gloves on for this. Your screen may have been done by automation. And because this is skipping serologic work, this is often also called, at least locally, cross-match on demand. The facetious alternative is uncross-matched blood. You may be asked, how many ways are there to cross-match? You can appreciate that Coombs phase complete cross-matching is the slowest of all techniques because of the incubation requirement, and that immediate spin, electronic, and uncross-matched blood are higher and higher speed alternatives. How do we pick platforms to investigate antibodies within? Well, these are choices made by labs, and sometimes within labs if they have multiple platforms available, depending on what the circumstances are. Tube testing uses the most of your patient's sample, and the solid phase microtiter plate the least. Solid phase microtiter plates are notoriously oversensitive, and so there are many method-dependent antibodies that assail this technology. But at the same time, solid phase picks up more antibodies than what might otherwise get picked up or therefore missed in tube or gel. You have freedoms when you work in tube as well as in gel microtube in terms of fiddling with enhancers or altering your cells. We have automation for gel and solid phase microtiter based testing. And in terms of the visual readouts, which were shown previously, what does a positive versus a negative look like? The inter-observer agreement is very high in robot recognized endpoints in automation, but also at a manual level when working in the gel microtube technique. You get a lot more inter-observer variation when two serologists are looking at the same test tube reaction. The advantage of tube testing, as alluded to previously, is that you've got these degrees of freedom. You can examine a panel at immediate spin. This is going to be picking up your IgM repertoire. You can heat it up to 37 degrees before adding your Coombs reagent to see if the IgMs that you've identified at immediate spin have high thermal range, in which case this might be an explanation or a confounder for your anti-human globulin test. So this is essentially showing what you might need to be subtracting for in your understanding of your anti-human globulin reaction. But finally, your anti-human globulin reaction is your 37-degree incubation with your anti-human globulin to determine truly whether or not there are IgGs involved. And these are the more clinically significant in vivo antibodies when it comes to the protein-based systems that these antibodies are targeting. This is showing what the semi-quantitative scale looks like in gel microtube technique. And what you have happening very nicely in the microtube is the clarity of the mixed field. This is not an ambiguous result. And so many papers illustrating mixed fields will often use the gel microtiter plate to prove this assignment in this very clear visual way. The solid phase microtiter plate technology uses an antigen-coated well with low ionic strength saline in your patient's plasma or serum. There are indicator red cells that have Coombs reagent attached to them. After an incubation and washing, the proofing of whether or not your patient has antibodies binding to that cell of interest is going to be an indicator red cell that has Coombs reagent. If the Coombs reagent is interacting with FCs reflecting patient-bound antibody to the reagent red cell surface, there will be a red coating illustrating the binding of the anti-human globulin. It is, again, bound to indicator red cells, and that will give your blush or your red hue to reflect the positive response. Now, what if you have a positive screen emerging during an urgent need for blood? Well, you have to contemplate the risks of harm. An O-neg red cell may not be more likely to skirt an antibody than type-specific blood. So doing something incomplete can sometimes be less harmful than deferring perfection. You've got to look at your patient and the testing timelines. What are the odds of having antibodies? That's going to relate to the risk of transfusing emergency released blood versus what is the likelihood of tolerating the anemia, which is going to speak to the risk of not transfusing that emergency release unit. So in other words, a spectrum exists for the risk of a hemolytic reaction from incomplete blood bank preparations. If this young, never transfused male is shot next to another gunshot wound victim, a middle-aged G6P6 female born with sickle cell disease and unsuccessfully bone marrow transplanted 20 years ago with 800 red cell transfusions under her belt and multiple red cell alloantibodies and low-grade chronic warm autoimmune hemolytic anemia, clearly the lady on the right is going to be imperiled by her emergency release on cross-match blood compared to the guy on the left, whose odds of having a positive screen are incredibly low, nearly zero. So emergency release considerations, the blood requirement urgency, exceeding the testing clearance time, speaks to how long it takes to group your patient, how long it takes to screen. The result of that negative screen takes you to a 1-hour finish line if you declare that specimen to be stat. You're out of luck, however, if your patient's screen is positive, because it can take longer than that hour to then deduce the specificities. And if there are multiple antibodies or a high prevalence antibody, then it can take quite a bit of time to source antigen negative blood. If you are screen positive, there's also the matter of having to run a complete Coombs phase cross-match to prove suitability of the intended unit. And so the theory here is that you can get to the finish line within an hour if you are lucky enough to be screen negative, but we can't promise that in all cases for those who are unfortunately found to be screen positive in this emergency situation. How long is a blood bank sample good for? 3 days, roughly speaking, although that's interpreted in different ways. UHN will interpret this as 4 days or 3 days after the day of draw, rounded up to that third day's midnight. That's what's considered a short interval. So low sensitization risk patients can be represented for longer. A sample, if you know that your patient has not been transfused or pregnant in the last 3 months, does not need to be repeated every 3 days. But the high sensitization risk individual needs refreshment of the sample because their immune system may be dynamic enough to be revealing different specificities by the time of that next test. How do you solve a panel? Well, let's take a look at 2 sample cases. Your patient's plasma is being examined against test cells. In the 2-cell screen, your patient may have antibodies against any of these classic structures of significance. In the 3-cell screen, you get a little bit more information. The 2-cell screen is usually an R1R1 versus an R2R2 cell, and the 3-cell screen has at its 3rd cell typically a little r. In the 3-cell screen, you can deduce an anti-D if the first 2 cells are reacting, but the 3rd one isn't. You can't do that in a 2-cell screen, but the 2-cell screen, given its antigram, can give you a few other insights or at least a differential diagnosis. And likewise, you have a little more power to do that with the deduction powers coming from a 3-cell antigram. So let's say your patient has antibodies. If they're glomming onto screening cell 1, it's not going to be an anti-D. But if screening cell 1 is the only one that's kill positive, well, it's a pretty good guess that this is the antibody at play. In this particular 3-cell screen, you could see that there was a hint of a reaction at 37 degrees, 1+, but not as strong as the reaction with the anti-human globulin. So this could be a new seroconversion with a little bit of residual IgM at a high thermal range that is mostly isotype switched to the IgG. You could see that in this mini panel or 3-cell screen that an anti-big C is also on the differential diagnosis because screening cell 1, the cell under attack, is also big C positive. So how do you sort out the difference? Well, you do a panel investigation. This gives you a more informative view. This is showing how we do rule outs. Cells that give you non-reaction outcomes are the ones whose antigen expressions give you a sense of what the antibody is not up against. If you had an anti-D, you would have expected a reaction, but you have no reaction here, and so D can be ruled out. Likewise, the fact that the cell is big E positive and little c positive means that these are doubtful to be targets. And so again, this is how you do rule outs. And in the last step, the little r cell has little c and little e ruled out, as well as a couple of other things like M. Now, because not all antigens have been ruled out, we cannot rule out the possibility of lower prevalence antigens. The ones in white are the antigens that none of the screening cells even expressed. So if a person has an antibody, we cannot rule out that they don't have an anti-CW or an anti-V on the basis of these screens, even if they were completely negative. They just weren't there to detect what antibodies your patient may have had. The blue arrows are again showing what the leading suspects are for this reaction panel. You're really just left with big C or Kel as far as what you can potentially rule in. The hashed arrows here reflect slightly less certainty because we have the issue of homozygosity versus heterozygosity. In screening cell 1, if you have big S and little s, or single dose expression, it's harder to rule out or rule in whether or not an antibody is at play because there were fewer targets available. In this panel here, we can do our rule outs a lot more effectively. We can also just eyeball what lines up best with the positive reactions. You can see Kel is lining up beautifully, whereas big C isn't at this panel. And in 2x2, you can do this more formally or systematically. The p-value for the hypothesis of an anti-big C is 1.0. There are 2 cells that are quote unquote false positive, where not having big C nevertheless produces a reaction. And conversely, 2 instances where you express the big C that a reaction is not witnessed, so false negatives. This blows the hypothesis apart. But what works? The Kel idea. All 3 of 3 Kel positive cells manifest in a reaction, and all 8 of 8 Kel negative cells are non-reactive with no false positives and no false negatives. The p-value for this relationship is 0.006, so less than 0.05, statistically significant, and therefore endowing you with the power to conclude it's anti-Kel. You can do your home computation of p-value by the 2x2 table and this factorial calculation, or you can look at reference tables or use online calculators to establish your p-value in these 2x2 exercises. To get a p-value of 0.05 or less, you want to work with at least 3 antigen positive and 3 antigen negative cells. But there are other ways to get to a combination that gives you a significant p-value. If you have a high prevalence antibody, you can't as easily pick 3 and 3, but you may want to work with 6 cells that are positive and 3 that are negative, 7 cells that are positive and 1 that's negative. Don't quite get you there, but as you can see, there are other combinations that can enrich your confidence. What is dosage? This is whether you express an antigen homozygously, which is to say in double dose, versus heterozygously, which means in single dose. If you have antibodies to an antigen in single dose, you may not see a reaction, and you may miss the power to deduce the existence of that specificity. So in codominant systems, like the CE system, the KID antigens, the Duffy antigens, and the MNS system, you can miss antibodies, or you can see varying strengths in accordance with the dosage. So weaker reactions with single dose cells and stronger reactions with double dose cells. A few words on the KID blood group system. 2 alleles, JK and JKB. It's important to appreciate that KID antibodies can fix complement and therefore generate severe acute or delayed hemolytic reactions. Anti-JKA is implicated in 1/3 of all DHTRs. JKA is common, 28 plus 50, 78% of donors. And with anti-JKAs having one of the fastest evanescence kinetics of any antibody described, 1/3 disappearing in a week and 3/4 disappearing in 3 months, no surprise. The availability of the stimulus is high, and the disappearance of the antibody made against it very slippery. So be aware of kids. Very useful for patients to have antibodies in registries, or to at least bridge themselves with antibody cards, because patients are a lot more portable than their health information is. Now, how do you make sense of an inscrutable panel? What if you have all cells reacting? You have no rule outs. And so you're out of luck here unless you turn this into this. You can have new vision with clues on likely targets for culprit antibodies. If some reactions are cleaned up or eliminated, and you know what the reagent-sensitive systems are for that, versus other reactions being differentially strengthened by a reagent, you can get dualistic insights all at the same time. This is like having B vision. So what are these transforming reagents that give you rule out clarity and physical chemical clues as to the nature of the systems involved in the antibody repertoire? Well, enzyme treatment classically. Papain is what we use. You can also sulfhydryl treat with dithiothriotol or 2-mercaptoethanol. This will break sulfhydryl bonds. Those would be peculiar to the KEL system, most notably. ZZAP is a combination of papain with DTT. So this is a very strong way of eliminating numerous antigen systems. You can also do dilutions or titrations and plasma inhibitions, which, as I mentioned, are useful in inferring antibodies against soluble adsorbed antigen structures. In GLIAT, the gel microtube technique versus GLIAT using enzyme-treated cells or gens, you can classify antigen systems based on what enzymes do to them. Reactions that get enhanced in enzyme-treated red cells are your ABO and RHD, your 2 major systems and things related to those. So carbohydrate antigens, Lewis, big I versus little I, and ABO are examples of enhanced antigens. You're going to see more reactions or higher strength reactions with enzyme-treated cells if these are the targets. P is brought out, RH is brought out, autoantibodies are enhanced, and KID antibodies are also enhanced. Enzymes will eliminate the target of antibodies facing or cognate with the MNS system, PR or the protease system, the Cartwright system, the Duffy system, at least the flappy tail upon which the Duffy A versus B determinant exists, and the Scheiter-Rogers system. Again, that was one of our soluble antigen systems. Enzymes can obviously melt these non-intrinsic structures right off. KEL antibodies tend to be unaffected by enzyme treatment. There are some cute mnemonics here at the bottom to also help remember these. How do you distinguish between a high-frequency alloantibody and a warm auto? Well, let's say you have a warm auto and it is attracted to a public antigen, a universal antigen present on all red cells, hence the classic pan-reactive pattern, reacting equally with self and non-self. The warm auto reacts with all target cells as well as self. So this is going to be your clue. Otherwise, a high prevalence antibody will react with all cells except for one self. So the autocontrol and its relationship to the rest of the panel is incredibly important in making such a distinction. No surprise if you are incompatible with yourself and every panel or reagent red cell. You will also be incompatible with a segment from an intended unit, no matter how supermatched it is. Even if you were running a cross-match with your patient's plasma against their own induced pluripotential stem cells cloned up in a bioreactor because they're currently incompatible with themselves. Even an ex vivo grown cohort of their own red cells is obviously going to be incompatible. The testing against or of self comes in two formats, the autocontrol or the DAT. The autocontrol is quite a bit more sensitive. You are reuniting the patient's own red cells with their own plasma. It's an incubated technique, and there are usually enhancing reagents. And that's in contrast with a tube DAT, where the individual's red cells are washed, the testing is done at room temperature, and there are no enhancing reagents. So this explains why autocontrols are sometimes more positive than the DAT. The DAT can be negative in 10% of immune hemolytic anemias, but where that warm auto may show up is on enzyme-treated cell panels or on autocontrols. Terminologically, the adsorption is different from the eluate. The adsorption is about cleaning up a mess and trying to see beneath a repertoire of autoantibodies when you're working in the plasma phase. How do you tell if they're alloantibodies? Well, you need to do an adsorption if you're faced with an obfuscating repertoire of autoantibodies. An eluate is about peeling off what's bound to the patient's red cells and studying exactly what it's binding to by running that new suspension against reagent red cells. So the emphasis of an eluate is informing what we know of the patient's current red cell attack story. So here's a suspect screen that is actually positive due to a mix of auto and alloantibodies. There's an anti-Kel with a warm auto, but how do you tell that an anti-Kel is there if all the reagent red cells are reacting equally under the influence of the warm auto? Again, the autoantibody is masking the detection of the anti-Kel. You just can't see that reaction. So the autoadsorption is a solution. Your patient's red cells can only adsorb out the autoantibody. Alloantibodies, by definition, are antibodies against non-self. So using your patient's erythrocytes as a repetitive rebinding surface to clarify their own plasma is the adsorption maneuver that will enable you to eventually see the anti-Kel. And indeed, this is exactly what we're seeing in this panel investigation. Now we can see the anti-Kel reactions, no problem. You cannot do an autoadsorption, though, if your patient was recently transfused, because the transfused red cells, which may be bearing the alloantigen to which your patient has alloantibodies, may be repetitively binding those out in your adsorption cycles. And now you're left with nothing. This clarified plasma is over-reduced to the point of false negativity. Adsorptions, again, shown here, are treating the patient's red cells to clean the surface up so that you've got more binding sites available. Once you've cleaned up a sample of the patient's red cells, they reunite with the plasma to bind out the antibody. And then that serum, which is removed, is now testable and informative. If your patient has been transfused, you can do what's known as a differential or allogeneic adsorption. You can use screening cell 1, screening cell 2, and screening cell 3 type red cells, R1, R1s, R2, R2s, and little r. Yes, you might adsorb out an anti-big C or an anti-little e in the first system, but because screening cell 2 is antithetical, that cell won't bind out the antibody. And so each of these plasmas are differentially going to reveal different things. And between the three of them, you are hoping to reconstitute your patient's truth in the semi-informative nature of each resultant clarified plasma. The alternative to a differential allogeneic adsorption is to select a cell type that is as close to your patient as possible. It can be phenotype or genotype informed. So if you use that cell, it may be a good enough surrogate of your patient's red cell type. The extent to which it is a supermatched representation is the extent to which it can safely adsorb out nothing but the autos and give you an informative alloantibody discovering adsorbate. Determining your patient's red cell profile also helps to verify the antibody specificity that you've picked up on your panel. If your patient has made an alloantibody, the patient, him or herself, should be antigen negative for what they've made an antibody against. The identification of an anti-Kel can be validated by proving that your patient is Kel negative. If the patient has never been transfused, this is the prime opportunity to establish that proof or to have your patient's profile make sense in the context of the antibody that they've made. So this is an adjunct in confirming alloimmunization. And once you've phenotyped your patient, you enhance the opportunity to select transfusions that are better matched. So phenotyping more broadly allows you to get pickier by virtue of that knowledge. If you are matching at a higher level the warm autoantibody patient and you don't know that they may have alloantibodies reactivatable or present currently, while your adsorption is still cooking, you can bypass historic antibodies and also prevent the formation of new ones or primary sensitizations, which patients with warm autoimmune hemolytic anemia are probably twice as likely to do as patients without warm auto. The notion of compatibility speaks to whether or not you are bringing to the patient a phenotype similar unit or proving that their serum cooperates with the candidate red cell unit in an actual serologic cross-match. These are complementary notions, but sometimes compatibility is spoken to by only one act or the other. So it is helpful to be clear in communication on what you mean by this. Now, what if you have an unidentified antibody on a panel or a cross-match? Sometimes you run a panel, you've ruled everything out, but you still don't have an explanation for that smattering of reactions, be they just a few or many. The differential diagnosis for the unidentified antibody is an artifact. It could be a low frequency antibody, but then you're stuck with figuring out what its significance is if you have a name for it or not. This is where there may be a role for monocyte monolayer antibody testing. The monocyte monolayer assay can, irrespective of whether or not you know the specificity of that antibody, tell you if the unidentified antibody is clinically significant or not. If the antibody induces phagocytosis, you have a problem, as opposed to if there is no phagocytosis induced by that antibody attaching to the candidate red cell. The vital signs done at 15 minutes, par for the CSA standards, will also be a clue. This is, in essence, a test of in vivo compatibility. In the first 15 minutes, you're not transfusing a lot of blood. If you're giving that test dose at a rate of 50 mils per hour, and you're checking in at the 15-minute point, you can't have infused more than 12.5 cc's minus a bit of dead space. And so that little head of transfusion, if it's an incompatible erythrocyte aliquot, will typically provoke fever as the first sign of incompatibility. And so anyone who's beginning to react after such a scant exposure is a major red flag that maybe we should be cutting out. That may be the early proof of incompatibility, in other words. The other possibility for an unidentified antibody is a BG, or an antibody against HLA. Now, we appreciate that red cells don't express HLA intrinsically across the red cell surface, but they may adsorb soluble HLA. And if there are HLA antibodies in a heavily presensitized host, well, those may land on those soluble HLA antigens. Some passive antibodies may also be the cause of an unidentified antibody. The parallel in hematology, what I'm saying here is that the nuisance of the unidentified antibody is analogous to the conclusion of nonspecific inhibitor in the coagulation lab. So BGs, these are the HLA antibodies. BG is the acronym for Bennett-Goodspeed. There are numerous BGs, but BGA, BGB, and BGC reflect more common HLA specificities. You can see what the population frequencies are here, and you can see that these got recognized in their order of population relevance. These, again, are not clinically significant. They're like the Shido-Rogers or the Lewis. They are simply adsorbed antigens, but they do slip off, and so they are unlikely to be the foundation of a hemolytic transfusion reaction. Thankfully, BG positive cells are often excluded from screening cells, but some panels have BG positive cells, and the specific attribute therein is sometimes helpfully revealed on the antigram, so that if the unidentified reactivity lines up with those BG cells, well, then there's your answer. Now, a few words on the monocyte monolayer assay. This is not just about mingling your patient's plasma with the red cells to see if antibodies are binding. This is to see if peripheral blood mononuclear cells recognizes that binding as a binding of phagocytic interest, or an ex vivo living cross-match. You can permute your experiments in different ways. You can use as your red cell substrate self or non-self red cells. The non-selfers could be already transfused blood if you're trying to figure out if this was incompatible in retrospect, or a candidate unit whose compatibility you really want some assurance on. The opsonin is typically your patient's serum or plasma, but you can sub in an albumin control as another element in this experiment. And in terms of your peripheral blood mononuclear cells, you can use your patient's own mononuclear cells, which may be in a state of macrophage activation or cytokine storm and more prone to phagocytosing than the use of allogeneic cells. If you have this signal, well, you can make some conclusions on so-called additional lesions. Your patient may have a hemolytic antibody, but their phagocytes may be particularly hungry, and that can make for a double jeopardy. And so this kind of experimentation can really disambiguate what is the lesion and where is the lesion. As mentioned by those substitution or choice options, a 2 erythrocyte, 2 monocyte, 2 opsonin model already gives you 8 different experiments, and they are all going to give you different insights or answer different questions. If you are using peripheral blood mononuclear cells from allogeneic controls with an antibody-free material and the patient's own red cells, this is simply asking, do healthy monocytes eat up a patient's erythrocytes in an antibody-free environment? This is answering the question of whether or not the patient's own red cells are eryptotic, for lack of a better word. In the next experiment, if you are running this system against allogeneic red cells, do healthy monocytes eat up a candidate unit in an antibody-free environment? That would not be expected because healthy donor cells are less likely to be eryptotic than your own patients. In this other experiment, do patient monocytes, this is what's subbed in here now, eat up their own red cells in an antibody-free environment? In this next experiment, do the patient's monocytes eat donor erythrocytes in an antibody-free environment? Is the immune ramp up so high that an allogeneic red cell is indiscriminately being consumed? In this experiment, are the patient's own red cells in her own plasma being phagocytosed by normal monocytes? Is this a warm auto that would be happening in the immune tone of another immune system if those were the phagocytes at hand? Are donor cells in the host's plasma capable of inciting phagocytosis by normal monocytes? Are the patient's own red cells in her own plasma being phagocytosed maximally by her own monocytes? Is this the triple jeopardy? And finally, are donor red cells in this patient's plasma being phagocytosed by their monocytes? A very useful answer, the strongest model for a potential in vivo incompatibility. A warning: the mechanics of the MMA So a Ficol separation will help isolate the mononucleocytes, the monocytes for the adherence assay. And on this slide, each of these little squares represents each of the experiments. And so this is an 8-well system, which means you can do all of the foregoing experiments shown on the previous slide. The yellow stuff is mingled with the red stuff, and the monocyte layer is then watched for how much phagocytosis is happening. The number of red cells phagocytosed in every 100 monocytes gives you your phagocytic index, and a value greater than 5 is considered significant. Here are some pictures of the assay. Attachment may be a precursor or a predictor of hemolysis, but the most significant predictor would be phagocytosis, so actually seeing the erythrocytes within each phagocyte. So to conclude, we have covered the nucleus, grouping being about what blood type to transfuse your patient with at the most important decisional level. The screen about the non-ABO antibodies that could threaten a forthcoming unit despite it being group suitable per the kernel principle. The theoretical snapshots surmised by informative group O testing cells. And finally, cross-matching. Is the proposed unit compatible? Thank you for listening.